Welcome to this lecture on the basics of neuroscience. Today, we are diving into the fascinating world of neurotransmitters. If you have ever wondered how a thought forms, how we feel happiness, or how we command our muscles to move, the answer lies in these tiny chemical messengers. By the end of this session, you will understand exactly what they are, how they are categorized, and even the mathematics behind how they bind to our cells. Let us begin our journey into the microscopic world of the brain. To start, let us build an intuitive understanding of how neurons communicate. Imagine you are standing on one side of a rushing river, and your friend is on the other. You cannot shout over the noise, and there is no bridge. Instead, you write a message on a paper airplane and throw it across. In the human brain, neurons face a similar problem. They do not actually touch each other. They are separated by a tiny gap called the synaptic cleft. To send a message, the sending neuron releases chemical paper airplanes that float across this space and land on the receiving neuron. These are neurotransmitters, and without them, our brain would be a collection of silent, isolated cells. Now that we have a conceptual picture, let us look at the strict, formal definition used by neuroscientists. A neurotransmitter is an endogenous chemical messenger that transmits signals across a chemical synapse. But not just any chemical qualifies. It must meet rigorous criteria. First, it must be synthesized and stored within the presynaptic neuron. This means the sending cell makes the chemical and keeps it ready in tiny bubbles called vesicles. Second, it must be released by the presynaptic axon terminal specifically in response to an electrical signal, which we call an action potential. This ensures that the chemical is only released when the neuron intentionally fires. Continuing from our previous criteria, there are two more strict rules a molecule must follow to be called a neurotransmitter. Third, if we artificially apply this chemical to a postsynaptic cell in a laboratory, it must produce the exact same response as the naturally released chemical. This proves the chemical itself is responsible for the message. Finally, there must be a specific mechanism to quickly remove it from the synaptic cleft once the message is delivered. This happens through processes like enzymatic degradation or reuptake back into the sending cell. This rapid cleanup is crucial, so the signal does not last forever and overwhelm the receiving cell. Understanding these criteria leads us to why neurotransmitters are absolutely critical in the real world. They govern every single aspect of human behavior and physiology. When you lift your arm or when you feel happy, specific neurotransmitters are at work. If we did not have distinct types of these chemicals, our nervous system would just be a simple on or off switch, making complex thought impossible. In modern medicine, almost all psychiatric and neurological medications work by targeting these specific systems. For example, antidepressants often work by boosting the levels of serotonin, while treatments for Parkinson's disease aim to replace lost dopamine. They are the true master switches of our biology. Now that we know why they are important, let us categorize them. Neurotransmitters are generally grouped into three main chemical categories. The first category is amino acids, which are the workhorses of the brain, responsible for most fast point-to-point communication. The two most famous are glutamate and GABA. Glutamate is the primary excitatory neurotransmitter. When it crosses the synapse, it tells the next cell to fire its electrical signal. On the other hand, GABA is the primary inhibitory neurotransmitter. It acts exactly like a brake pedal, telling the receiving cell to stop firing. Together, they balance the brain's overall electrical activity. Moving beyond the fast-acting amino acids, we have our second and third categories. The second category is biogenic amines. This group includes famous molecules like dopamine, serotonin, and norepinephrine. Instead of just sending fast point-to-point signals, amines usually modulate broader brain states, such as your overall mood, your level of arousal, and your sense of reward. Finally, we have peptide neurotransmitters. These are much larger molecules. A great example is endorphins, which act as the body's natural painkillers. They are often released during intense exercise or stress to help us push through pain. Each category serves a highly specialized role. Before we move to the mathematics of how these chemicals bind, we must mention one very special neurotransmitter that stands in its own unique chemical class: acetylcholine. It does not fit neatly into the amino acid, amine, or peptide categories. Acetylcholine is incredibly famous for two main jobs. First, it is the primary messenger that controls muscle contractions at the neuromuscular junction. Every time you move a muscle, acetylcholine is bridging the gap between your nerves and your muscle fibers. Second, within the brain itself, it plays a crucial role in supporting memory formation and learning. It is a highly versatile and essential molecule. Now that we understand the types of neurotransmitters, let us look at how effectively they work using mathematics. This is described by the fractional occupancy equation, which tells us what percentage of receptors are currently activated. The formula is Y equals L divided by the sum of L and K sub D. Let us break down these symbols. Y represents the fractional occupancy, a number between 0 and 1 indicating the proportion of bound receptors. L represents the concentration of the ligand, or neurotransmitter, floating in the synaptic cleft. K sub D is the dissociation constant, which is the concentration where exactly half the receptors are occupied. Conceptually, as the amount of neurotransmitter increases, more receptors get filled. If K sub D is high, the neurotransmitter has a lower affinity, meaning you need a much higher concentration to get the same biological effect. Let us apply this formula with a concrete example. Imagine a synapse in the hippocampus releases glutamate, reaching a peak concentration of 10 micromolar. The receptors have a known dissociation constant of 2 micromolar. We want to find the fractional occupancy to see how strong the signal is. We are given the ligand concentration and the dissociation constant, both in micromolar, so no unit conversion is needed. We substitute 10 for L and 2 for K sub D into our equation. We first add the denominator, 10 plus 2, giving us 12. This represents the neurotransmitter competing with its own binding affinity. Finally, dividing 10 by 12 gives us 0.833. Physically, this means roughly 83.3% of the receptors are bound, creating a very strong excitatory signal likely to trigger an action potential. Now let us look at a clinical example involving Parkinson's disease, comparing natural dopamine to a synthetic drug. First, we calculate the occupancy for the natural neurotransmitter, dopamine. We are given a concentration of 10 nanomolar, and dopamine has a dissociation constant of 50 nanomolar at this specific motor receptor. We plug 10 into the numerator and denominator and insert the dissociation constant of 50. Adding the denominator gives us 60. Dividing 10 by 60 yields a final value of 0.167. This means only about 16.7% of the receptors are activated. In a physical sense, this is a relatively weak signal that might not be enough to trigger proper muscle movement, perfectly illustrating the deficit seen in Parkinson's patients. To help this patient, we introduce a synthetic dopamine agonist drug designed to be much stickier. We will calculate its occupancy at the exact same concentration of 10 nanomolar. However, this new drug has a much lower dissociation constant of just 10 nanomolar. We set up the exact same equation, replacing the K sub D value with 10. The denominator becomes 10 plus 10, which is 20. Dividing 10 by 20 gives us a clean value of 0.50. This means exactly 50% of the receptors are activated by the synthetic drug at the same concentration. Physically, this shows the drug has a much higher affinity, activating 3 times as many receptors as the natural chemical, which is why it is highly effective at restoring lost motor function. Now that we have calculated how these chemicals bind, let us visualize the actual physical space where this happens. On screen is a diagram of synaptic transmission. At the top, you see the presynaptic terminal, which is the end of the sending neuron. Inside it are circular synaptic vesicles, which are the storage bubbles filled with neurotransmitters, shown as small orange dots. When an electrical signal arrives, these vesicles fuse with the membrane and release the neurotransmitters into the middle gap, known as the synaptic cleft. The neurotransmitters float across this space and bind to the Y-shaped receptors located on the postsynaptic dendritic spine at the bottom. This binding is exactly what we just modeled with our fractional occupancy equations. It is a beautiful, highly coordinated biological mechanism. We have covered a lot of ground today, so let us summarize the most important ideas. First, neurotransmitters are the essential chemical messengers that allow neurons to communicate across the synaptic cleft. Second, they come in various specialized types, including fast-acting amino acids like excitatory glutamate and inhibitory GABA, mood-modulating amines, and larger peptides. Finally, the effectiveness of these messengers depends heavily on their concentration and binding affinity, which we can mathematically calculate using the fractional occupancy equation. By mastering how these chemicals are released and how they bind, you now hold the key to understanding everything from basic muscle movement to complex neurological diseases. Great job following along, and keep exploring the wonders of neuroscience.